mRNA vaccines changed the world almost overnight, right? They saved millions of lives during the pandemic and completely kicked down the door for a new era of RNA therapeutics. But in the fast-paced world of molecular biology, scientists are already in the lab working on something even more powerful. We're talking about an evolutionary upgrade. Welcome to This Explainer. Today, we're exploring a molecule with no ends to degrade, one that offers incredibly long-lasting protein production and has the sheer potential to outperform mRNA itself.
It's a massive biological question to ponder. Could a seamless, microscopic ring of RNA actually replace the vaccines that save the world? To really understand why researchers are so incredibly hyped about this, we need to zoom in and look at the fundamental structural differences between these molecules. So think of traditional mRNA as a straight ribbon, or an open piece of rope. Because it's linear, it obviously has two exposed ends, and inside our cells, there's a whole cleanup crew of enzymes, exonucleases, that naturally attack those exposed ends, causing that RNA rope to fray and degrade incredibly fast. Circular RNA, on the other hand, is a totally different beast. It forms a completely seamless ring. Because there are literally no free ends for those enzymes to easily chew on, it totally resists that normal wear and tear.
Let's formally introduce the hero of our story here for the biology enthusiasts listening. Circular RNA, or circRNA, is a highly stable class of RNA characterized by a unique closed-loop structure. The 5' and 3' ends are covalently linked together. By entirely hiding those ends, the molecule becomes a fortress against that exonuclease degradation we just talked about. It survives inside the chaotic, hostile environment of a cell far longer than linear RNA ever could.
Now, you might be wondering, just how much longer does it actually survive? Well, to put that into perspective, in vitro transcribed circRNA has been empirically estimated to have a half-life of 56 to 86 hours. Compare that to traditional mRNA, which has a half-life of merely 15 to 24 hours, depending on the cell type. That is a game-changing leap in intracellular stability. Okay, let's really dive into this. The implications of that massive stability boost are so profound that a recent paper in Nature Reviews Genetics made a pretty bold claim. They stated, circRNAs may eventually outcompete mRNAs as the preferred RNA format for clinical development. Those are massive stakes. We are quite literally watching a new scientific race unfold right before our eyes. Because circRNA sticks around for so long, it acts like a relentless molecular factory. The ribosome, our cell's protein-making machinery, can just keep going around and around the loop, almost like a race car on a track. This extraordinary intracellular stability results in a continuous, prolonged expression profile, which yields a 3 to 5-fold benefit in protein production over traditional mRNA. That translates to higher efficacy and potentially a massive reduction in treatment costs. Section 1. The next generation of vaccines and prolonged antigen production. So what does this all mean for your immune system? Well, vaccines work by producing antigens that train the body to fight off invaders. Because circRNA stays active much longer, it continuously produces those antigens. In models testing against pathogens like SARS-CoV-2, this prolonged expression trained the immune system to build a significantly stronger, more potent shield. Now, biochemically speaking, because circRNA is a closed circle, it lacks the 5' cap that linear mRNA relies on to kickstart translation. So it has to use something called an internal ribosome entry site, or IRIS, to get the engine running. But once it gets going, that continuous production could allow for what we call dose sparing, meaning patients might need a fraction of the usual dose, leading to way less toxicity and fewer side effects.
Section 2. Making circular RNA and engineering the loop.
Shifting gears a bit, how do scientists actually manufacture these microscopic rings in a lab? To create circRNA in vitro, they generally follow a specific biochemical recipe. First, they transcribe a linear RNA precursor. Then, they have to essentially trick that open piece of RNA into closing its own loop. They can achieve this through enzymatic ligation, utilizing tools like T4 ligases to glue the end shut, or they can use a mechanism that's arguably even more fascinating, ribozyme-mediated self-circularization.
What's absolutely wild is how that self-circularization process works. Specifically, the PIE and STS systems act as self-contained catalytic factories. Using group 1 iontrons, they execute a precise chemical cut-and-paste job. 2 transesterification reactions to be exact, splicing the RNA ends perfectly together without needing any extra ligase enzymes added to the mix. It's essentially molecular self-assembly.
Section 3, hurdles to overcome. It's time for a reality check.
Now, we have to be completely honest here. Every great biological breakthrough hits severe bottlenecks before ever reaching the clinic. And the reality is, synthesizing highly pure circRNA at an industrial scale is incredibly difficult. A massive hurdle right now is purification. You have to perfectly separate the functional circular molecules from all the leftover linear pieces and nicked circles. Scientists frequently use an enzyme called RNase R to chew up the linear scraps, but getting a perfectly pure batch is technically agonizing. On top of that, those elegant self-splicing methods we just discussed can unfortunately leave behind tiny foreign sequences, molecular scars that might trigger unwanted immune responses. And finally, just like the hurdles we face with mRNA, efficiently packing these rings into lipid nanoparticles, or LNPs, to deliver them safely into target tissues requires a ton of ongoing optimization.
Section 4. More than just vaccines. Expanding the clinical horizon.
Despite those hurdles, researchers are pushing forward relentlessly because the potential payoff is just staggering. Vaccines are literally just the tip of the iceberg for this technology. Beyond churning out proteins, these durable little loops can perform incredible non-coding functions inside our cells. They can act as molecular sponges, soaking up and sequestering disease-causing microRNAs that drive things like tumors. They can serve as protein scaffolds, physically bringing specific targets together for degradation. They can even be used as incredibly stable guide RNAs, making CRISPR gene editing exponentially more efficient.
Let's see how all this builds into an unbelievably diverse preclinical pipeline. We are already looking at LNP-packaged circRNA encoding CAR T cells to hunt down B-cell lymphoma. We're talking about durable gene therapies for cystic fibrosis. We're looking at deploying those mRNA sponges to treat severe heart conditions like cardiac hypertrophy, and even guiding ADAR1 enzymes for highly precise, targeted genetic editing. The therapeutic versatility here is just boundless.
So, to bring it all together, linear mRNA vaccines proved to the entire world that RNA could be deployed as a safe, programmable medicine. But circular RNA, which is more stable, longer-lasting, and potentially far more effective, is perfectly positioned to completely define the next era of RNA medicine. It takes all the hard-won lessons from the pandemic and evolves them into a durable molecular chassis that could conquer cancer, genetic disorders, and so much more. We are undeniably standing at the edge of a completely new medical paradigm. The question is no longer whether circRNA is promising. The data absolutely proves that it is. The real question is, how soon will it become reality? How soon will these seamless, microscopic rings jump from the laboratory bench into the hospitals and pharmacies that serve us all? Thank you so much for joining me on this explainer to explore the very cutting edge of biology.